Hello everyone, welcome back to my channel Anytime Medicine, and today we are going to learn about gluconeogenesis. As the word implies, gluconeogenesis means formation of glucose. So without further ado, let's get started. Before that, subscribe to my channel so you cannot miss my future videos. So glucose can be synthesized from different structures like pyruvate, lactate, amino acid, propionate and glycerol that we are going to talk about each in this video. So this is the whole reactions of gluconeogenesis, but first we are going to talk about is pyruvate. How is it converted into oxaloacetate and it is converted into phosphoenol pyruvate. From that it will make the glucose. So pyruvate usually converted into acetyl coenzyme A, and that will go to TCA cycle, or it can go to gluconeogenesis via reverse process of glycolysis. If there is a low oxygen level, it will make the lactate, and that will go to liver and make glucose, that is called Cori cycles. Also it get converted into alanine in the muscles, and that will go to liver and make glucose, and that is called alanine cycle that we have talked about in glycolysis video. So if there is a high level of ATP, it will block the certain mediators in TCA cycle, and because of that acetyl coenzyme A level gets built up, and high level of acetyl coenzyme A will induce the pyruvate carboxylase enzyme, and it will drive the process of gluconeogenesis. So you have to keep in mind that acetyl coenzyme A will activate the pyruvate carboxylase enzyme, and it will drive the gluconeogenesis. So now we'll see how pyruvate get converted into phosphoenol pyruvate. First, pyruvate will converted into oxaloacetate with the help of pyruvate carboxylase enzyme. It will use ATP and carbon dioxide in the process. Pyruvate carboxylase enzyme's cofactor is biotin that we have seen in vitamin B complex video. Then oxaloacetate will convert it into phosphoenol pyruvate. In this process, it will use phosphoenol pyruvate carboxylase kinase enzyme, and it will use GTP. This pyruvate getting converted into oxaloacetate process occur in mitochondria, and from oxaloacetate to phosphoenol pyruvate, this process is occur in cytosol. Oxaloacetate will get into cytosol from mitochondria via malate shuttle. You have to keep in mind that oxaloacetate will get into cytosol via malate shuttle. So this is a rare syndrome which is pyruvate carboxylase deficiency. In this, child will failure to thrive because they cannot make new glucose, as they cannot do gluconeogenesis. So elevated pyruvate will make lactic acid, and it will lead to lactic acidosis because other pathways has shut down, that we are going to see how in next slide. As we have seen, if there is a high level of ATP, it will block the certain mediators of TCA cycle, and because of that there will be high level of acetyl coenzyme A. High level of acetyl coenzyme A will drive the gluconeogenesis by activating the pyruvate carboxylase enzyme. If we get deficient to pyruvate carboxylase enzyme, we cannot do both of these process. Therefore, it will lead to lactic acid formation by go to Cori cycle. It can go to alanine cycle too. Because of that there will be high level of alanine too. So now we'll see this step of gluconeogenesis, which is fructose 1,6 bisphosphate into fructose 6-phosphate. So fructose 6-phosphate gets converted into fructose 1,6 bisphosphate via phosphofructokinase 1, and this is a process of glycolysis, and fructose 1,6 bisphosphate gets converted into fructose 6-phosphate with the help of fructose 1,6 bisphosphatase 1, and this is a step of gluconeogenesis. As we have seen in glycolysis, the phosphofructokinase 1 is the rate limiting step of glycolysis. In this, fructose 1,6 bisphosphatase 1 is also a rate limiting step of gluconeogenesis. It gets activated when there is a high level of ATP, and it will stop when there is a high level of AMP and fructose 2,6 bisphosphatase, which is a regulator of those two enzymes. As I said earlier, that fructose 2,6-bisphosphate is a regulator of those two enzymes. Fructose 2,6-bisphosphate will induce the phosphofructokinase 1 enzyme, and it will drive the glycolysis process. And it will stop the fructose bisphosphatase 1 enzyme, so it cannot do the gluconeogenesis process. So if its level gets low, it will do the gluconeogenesis, as it cannot stop the fructose bisphosphatase 1 enzyme. So now we'll see certain regulators of those two enzymes. Phosphofructokinase 1, which is for glycolysis, is induced by AMP and fructose 2,6-bisphosphate, as we have seen earlier, and which is inhibited by ATP and citrate. In contrast to that, fructose 1,6-bisphosphatase 1, which is for gluconeogenesis, induced by ATP and inhibited by AMP and fructose 2,6-bisphosphate. So now we'll see the last step of making glucose, which is glucose 6-phosphate into glucose. So glucose 6-phosphate gets converted into glucose with the help of glucose 6-phosphatase enzyme, and this reaction occurs in endoplasmic reticulum in the cytosol. This occurs only in liver and kidneys, and other organs will convert glucose 6-phosphate into glycogen. They don't convert glucose 6-phosphate into glucose. So as I have mentioned earlier, that amino acid, lactate, acetone and glycerol also can make glucose. Now we'll see how amino acid will convert it into certain mediators of TCA cycle or directly into pyruvate, and that will go to glucose making process. Lactate gets converted into directly pyruvate and acetone too. Glycerol will convert into glyceraldehyde 3-phosphate, and that will go to glucose formation into gluconeogenesis. That's how all the structures gets converted into glucose. So thank you everyone for watching this video. If you like this video, hit the like button. Don't forget to subscribe my channel and share it with your friends. Thank you.